Thank you everyone for joining us for the Yale Department of Radiology Grand Rounds today. I am delighted to introduce Dr. David Mankoff from University of Pennsylvania. Dr. Mankoff did his undergraduate degree in physics here at Yale University, graduating summa cum laude and a member of the Phi Beta Kappa. He proceeded to obtain PhD and MD at the University of Pennsylvania as part of the medical scientist training program. He is board certified by American Board of Nuclear Medicine and is the Gerd Muehllehner Professor of Radiology. He is also the Vice Chair of Research at the Department of Radiology at the University of Pennsylvania and the Associate Director for Education and Training in the Penn Abramson Cancer Center. Dr. Mankoff's research interests focus on development of imaging technology and methodology and translation of novel tracers into clinical practice with specific focus on breast cancer. He is the principal investigator on multiple active NIH-funded grants. Thank you, Dr. Mankoff, for sharing your expertise with us.
Thank you, Miriam, and I'm really delighted to be back talking at Yale, at least virtually. I'm recognizing the libraries and many of your save screens. I think I'm probably spent a few late nights there. So New Haven and Yale and Pepe's Pizza hold a warm place in my heart, and I'm really delighted to be back for your grand rounds. So I'm going to go ahead and share my screen. And can you see that okay?
Yes, it is perfect.
Great. Okay. Thank you again for the invitation. I'm going to talk to you a little bit about FDG and beyond. And for many of those in the audience that may interpret FDG PET scans as part of what you're doing on daily business, you may be doing that while you're watching the talk. I warned Miriam, I might make your head spin a little bit, so you have to think a little bit about what you're looking at. I do, in particular, want to go through the acknowledgements so that I don't run past them in the end. And this has really been a number of folks from Penn and some of my former colleagues from the University of Washington and elsewhere. The folks who are highlighted in yellow have contributed directly to slides or the different approaches. As with the grants in yellow, I've been fortunate for 9 years. I just completed my stint to be a Komen Scholar. And so the Susan G. Komen has been an important part of funding this research, and I want to make sure they get credit. Okay, and if my slides advance here. Okay, I have some disclosures. None of these are going to be particularly relevant today, with the exception of material transfer from Calithera, a company that makes a glutaminase inhibitor, and Blue Earth Diagnostics that makes Flucycline PET tracer. I can't help but advertising a little bit for the Penn Molecular Imaging Program. This is many of the different programs we have in molecular imaging. There's a heavy emphasis on PET with a lot of strong tradition there, but also multiple modalities that include non-tracer based MR work, novel X-ray contrast using nanoparticles, radionuclide therapy run by our division chief, Dan Prima, along with Mehran Mekhandi and Sean Carlin, and an emerging optical and hyperpolarized type approaches. And today I'm really going to focus on breast cancer and want to acknowledge other folks that have worked with me closely on this that include Rong Xu, Austin Pantel, Sharon Lee, Hank Kong, Ravi Reddy, and others that you'll hear about. So I want to start by talking to you about imaging the glycolytic rate, or the glucose metabolic rate, with FDG PET. In the biochemical interpretation, it's not quite as simple as it would look from sitting down and reading PET scans all day. And then I want to go a little bit beyond fluorodeoxyglucose and glucose metabolism and talk a little bit about a biochemical framework and hypothesis and get to glutamine as an alternative fuel. I will save a little time at the end for the gadgets and talk to you a little bit about some novel approaches to four-dimensional analysis that have come out of parts of this effort, and some GWIZ stuff from our group, our physics group, which is where I did my PhD a long time ago, who have an operating total body PET, and I understand there will soon be one at Yale. Maybe there already. Just to remind those in the audience about PET, you start with a cyclotron in the beginning of the morning. Ours is down under the nursing school, not too far from where I'm sitting there right now. You go into a gleaming lab that is a hot cell that makes a FDA compliant tracer, in this case FDG. That comes out of the cyclotron, makes its way to the PET suite. And you can see the illustration that suggests I should not be a medical illustrator. I should keep my a day job. So you injecting something hot and red into a smiling patient, and then go into a PET CT scanner. Again, remembering that this PET portion of this is designed to be able to collect pairs of electron photon, the annihilation photons that result from electron positron interactions. And very importantly, you can get not only an image, but an image of absolute tracer quantification if you calibrated your scanner correctly. And you can make this as a dynamic image, kind of like the flow phase in a nuclear medicine study. And you can collect this information dynamically, even though that's not what we standardly do in the clinic. So here is FDG, which is probably, if you're doing clinical medicine, most of what you're doing in the clinic. If you talk to any of your friends that are chemists, or my friends are chemists, tell them I use the correct chair conformation in my diagrams. And just to remind you, FDG is a close analog of glucose. It is transported into cells in close analogy to glucose. It is phosphorylated by hexokinase, the rate limiting step in the early part of the path, just like glucose. But unlike glucose, it doesn't go further down the train of glucose metabolism. And in most tissues that don't have phosphorylase, including most tumors, FDG is trapped as FDG6P. And when you're looking at a scan, you're mostly looking at that FDG6P. And we'll come back to that, at least when you're looking at tumors. So this has worked really well. And in breast cancer, this has been very important for staging. I show you an example of a patient with a right locally advanced breast cancer, and has some mediastinal and internal mammary nodes. And if you're a good CT reader, you probably wouldn't have trouble finding that. But in some cases, they can be very difficult to find. And you can see how obvious this is on an FDG PET when you're using the metabolism to identify with great contrast the glucose metabolism, the aberrancy of the tumor compared to the rest of the body. This can also be very effective at measuring a response. This is something we presented at the RSNA a few years ago and are just about to send the paper out for the full study, where we can see response to therapy in this patient who is not on marrow-stimulating cytokines like GCSF, but actually has diffuse metastasis, had a great response to therapy. And you can see very quickly on this quantitative scale that this is responding quite well as early as 4 weeks. So this looks really good and works really well in the clinic. So thank you very much for inviting me, and that's the end of the talk. It's that simple. Well, it's not quite that simple, but before I go on and ruin your simplicity, not only is this pretty pictures, but in this case, in the same paper that this study came from, you can show that these measures are actually quite predictive of important outcomes such as progression-free overall survival and skeletally related events. Right, now it's not. I'm going to show you how it's not quite so simple. So first of all, what features are associated with tumor uptake in breast cancer, i.e. what are associated with glycolytic rate, or probably more correctly and generally glucose metabolism? So we know in human breast cancers, uptake is associated with high nuclear grade, and that may be in part because of the proxy of nuclear grade in breast cancer for the density of cells. The more cancer cells you have per milliliter, and the more they use glucose, the hotter it's going to be. We tend to find aberrant glucose metabolism in dedifferentiated tumors that are further away from the parent. In this case, these are the estrogen receptor or ER negative breast tumors, and in particular, the triple negatives that have lost estrogen receptor, progesterone receptor, and HER2. We also know that glucose consumption will vary with proliferation, at least in an approximate fashion, and the more rapidly proliferating tumors will have more glucose uptake. We also know certain things are associated with low uptake. A very well-differentiated histology, which is grade 1 lobular breast cancer. In breast cancer, may not even show up on an FDG PET-CT. It often has low nuclear grade, and those tumors that are ER positive will be less likely to have high uptake than those that do not. and as I showed you before, prior effective therapy. Now, Louis Chodosh, who's chair of our cancer biology and was my 4-year Yale College roommate, just to keep it as a Yale theme, has done some really elegant work in his genetically engineered mouse models and shown that a variety of oncogenes who drive these tumors will all contribute to the use of glucose that you can see in these micropet mouse models. However, they'll all contribute differently. Perhaps not surprisingly, for those that know these pathways, AKT and MYC-driven tumors are actually use a lot of glucose. Some of the things like RAS and NU and WNT use a little bit less. And again, this is not a surprise if you know these pathways and their activation of glucose, but it just goes to show you that there are multiple different gene pathways that can activate glucose. And so we're looking at this downstream signal. It's a little bit tough to tell exactly what we're looking at sometimes. So if that weren't complicated enough, when we look at FDG, we're not necessarily looking at glucose consumption. And this was really elegant work that was done by former colleagues at the University of Washington and the late Alex Spence, who compared carbon-11 glucose to fluorodeoxyglucose and showed that you could get a difference. The idea here is when this was first developed as a technique back at NIH by Lou Sokoloff and Marty Rivich, the idea was you could, from FDG, figure out what the glycolytic rate was in the normal brain. And as a matter of fact, it's a pretty good match. But the tumor doesn't listen to that. And in fact, the tumor has different enzymology. And when you're looking at FDG, you are looking at some measure of glucose consumption, but it's not exactly the same as true glucose. So head spinning idea number one. So now let me move you into some work that we did at about shortly after Alex and his group had finished up that work. And we took a little bit different look at this. And we looked at the perfusion of these tumors using O15 water. We also looked at the kinetics of glucose metabolism by looking not simply at a static version of FDG, but looking at a dynamic version of FDG. And the idea here was the understanding that perfusion is important and that this would allow us to look at water as a proxy for delivery of oxygen, delivery of substrates versus the substrate utilization. And in the end, we get the metabolic rate of FDG as a slightly more quantitative version of FDG metabolism than the SUV and a blood flow measurement from water. So what we found, probably wouldn't surprise those that know tumor biology, is that unlike normal tissues, which tend to have pretty matched glucose consumption and blood flow, this was all over the place in breast cancer. And as a matter of fact, this was a scattergram without any real correlation. You could have tumors of all kinds of different types, some with low flow, some with high flow, some with very high glucose consumption, and some without. So it's interesting, though, because these had, it turns out, importance in the study that we were looking at. So when you look at these pictures that are taken from the side, meant to look a little bit like an MLO view in mammography, this tumor at the top left corner of that diagram had very high glucose metabolism and relatively modest blood flow, and was way off axis there with very, very high FDG uptake that was out of proportion to its blood flow. On the bottom, there were some geographic differences, but quantitatively, there was a match that was a little bit closer to the normal breast. And actually, for this ER-positive tumor, it was actually fairly glycolytic, but had blood flow to match that. But these had important implications. The patient on the top had a great initial response to treatment, but the tumor never went away, and she unfortunately had early relapse and death. Patient on the bottom, which looked like maybe she didn't have quite aggressive and proliferative a tumor, responded actually quite well to systemic therapy and remains disease-free. So we're looking at two different phenotypes, both of which have some level of FDG uptake. And I'm going to now go and commercialize this a little bit and show you that we have the fuel guzzler that is chewing up gas as fast as it can, as fast as it's delivered. That's a Hummer for anybody who hasn't done that. And then on the bottom is the fuel efficient car that is a little bit more like what I think is a Honda Fit. Now, we went on a little bit later, and as we got more standard approaches to subtyping breast cancer, we noticed that there was a difference between the different subtypes. So in the ER-positive breast cancers, which are mostly luminal A or luminal B, if you're familiar with that, there was actually reasonably close correlation between the glucose metabolic rate measured by FDG and blood flow, And as a matter of fact, those tumors that had slightly higher FDG uptake that were indicative in that setting of probably the more proliferative end of the ER positive group, they actually responded well to chemotherapy. That's a complete response to treatment that you see in the solid lines. But on the right, in the extreme triple negatives, it looks a lot like that scattergram that I showed you, where this is all over the place. And in fact, many of the really hot tumors that were proliferative were also mismatched between their metabolic rate and their blood flow. Um, and in fact, this looked like a signal that suggested that some of these tumors with high uptake were less likely to respond than some of the ones with lower uptake and metabolism that were more matched to the blood flow. So if we go back here again, we've got the gas guzzlers that are kind of hard to treat, interestingly enough, and the fuel efficient tumors that have a little bit more expected pattern of blood flow tend to have a better response, at least in this series of patients who were treated with metronomic weekly doxorubicin and base therapy. So that's sort of where we stopped with FDG and water. And I will tell you, I've been chasing those results ever since to try to get a better explanation. And I think we're beginning to make some headway in understanding that there's life to FDG beyond, or there's life to metabolism beyond glucose, and not all glucose metabolism is glycolysis. So let me show you a little bit about that metabolic framework. So I have a very simplified diagram of glucose metabolism. To the metabolic specialists in the crowd, I apologize in advance. These will all be highly simplified. And the pathway that we are measuring is all the way over on the left, because FDG measures the rate limiting step here through the early metabolism of glucose, which is hexokinase. And so when something gets through hexokinase, we see increased uptake, and we can't really tell whether it's going down the glycolytic pathway to lactate or over to pyruvate. We'll come back to that at the very end of the talk and thinking about how other methods like hyperpolarized work might address that. And so these are very different fates, one of which is really just burning glucose rather inefficiently, and the other one which is utilizing the TCA cycle, and importantly, not only generating more energy, but has been pointed out by Craig Thompson, Chi Dang, both of whom were former cancer center directors here, that these molecules are also important for biosynthesis. And so we're going to look at a different molecule, glutamine, which is an amino acid, which can feed its way into the TCI cycle directly through the enzyme glutaminase or GLS. We're going to come back to those things a bunch of times in the talk. And so this is an alternative fuel supply in that approach. And we're going to look to see how that alternative fuel supply may or may not explain some of the findings that we just looked at. Now, I want you to think of this alternative fuel supply as different ends of the gas gauge. The stuff going down glycolysis is low octane. It's not generating a whole lot of energy from that. And the glutamine you can think of as the 93 high octane on your gas pump. And so the tools we have for these were developed by Hank Kung at the time when Craig Thompson was here. When Craig moved to Memorial Sloan Kettering, he took the fluoroglutamine with him and did some of the first in human studies in his lab, as I'll show you, some really elegant work. We also had carbon 11 glutamine, and I'll show you some of the work that we've been doing on that, which is interesting and tricky. But let's first focus on the fluoroglutamine. Fluoroglutamine is a very close analog of glutamine, and it is transported like glutamine. But actually, it turns out that it's not a great substrate for glutaminase, and it is not really metabolized to any great extent down to glutamate. There's a little bit, but not much. So this is a very different situation than what we're looking at with glucose. And as a matter of fact, I'll show you that fluoroglutamine is representative of other amino acid imaging agents that you've probably used in the clinic. And it's important to understand in this context how they work. Again, I mentioned that Sloan Kettering did some very elegant work in this space that first published at a Science Translational Medicine article in the 2015 work looking at glioma, showing that they had high uptake of fluoroglutamine in gliomas, much more so than some of the FDG studies, and had this case example of a patient whose recurrent tumor showed FDG in part of it, but didn't show FDG in the other part of it that was likely more glutamine transporting, at least in this case, and possibly more glutamine consuming. So it's really an early demonstration that you can get some different information by looking at this alternative fuel source than you can get by simply tracing the FDG pathway. We've been looking at this from a slightly different fashion, and in particular, we're interested in this molecule glutaminase that I mentioned before. Sorry, with my wrong button. And glutaminase is important because it's a target of treatments, including a company made by Calithera called CB839, which has been out in clinical trials. And the idea is if you could block glutaminase, and there are selective tumor glutaminase inhibitors, you could keep that tumor from using glutamine, and if it's addicted to glutamine, you might actually kill the tumor. And so this is really a lot of what I'm going to show you for the next 20 minutes or so about what we've done to study this. And again, here we are actually looking at the glutamine pool size as an indication of what this tracer traces, because it's not metabolized by GLS. So this is worth pausing for a second and taking you through a little bit of kinetics. And at the bottom, I've drawn diagrams for transport of these tracers into a compartment, for those used to compartment models, that's inside the tissue, and then the different fates of that tissue through the rate-limiting enzyme, either hexokinase for glucose or glutaminase for glutamine. Now, as I mentioned, when we look at glucose in active tumors, we're really looking at glucose 6P or FDG 6P is what we see on our scan. And so we see an early transport in parallel to blood, and then the tissue keeps rising, and we get very good contrast from tissue to blood. On the other hand, when we have these non-metabolized tracers of glutamine, for example, our fluoroglutamine, we're not getting to the substrate. We're not getting to the product. We're imaging the substrate. And so in imaging the substrate in those areas that are actively metabolizing the tracer, in all areas in general, we'll see reversible transport. And in fact, if you look at the diagrams on the right, unlike FDG, where the enzyme is active, and we see more of the product in the end of that, we see increased FDG uptake. It's exactly the reverse when we're looking at the substrate. So when we see tumors that have active glutaminase, our hypothesis is we're going to see less fluoroglutamine because it's distributing in a smaller pool, and if we wait long enough, we'll have longer uptake. For those that do amino acid imaging on a daily basis, if you've ever done a flucycline for prostate, you can't let that wash out too long. You have to image pretty quickly afterwards. And again, this is this same effect with a kinetic explanation. So does this hypothesis work? Well, this is really nice work from Charles Manning, who was then at Vanderbilt and now leads the molecular imaging effort program, or at least the chemistry program at MD Anderson. And there they used a transporter inhibitor of glutamine. And when you have that transporter inhibitor, oops, kept hitting the wrong button, you have slower uptake and you have a lower pool size. You have less rapid transport of glutamine into the system. And because you're still using that glutamine downstream when it's available, you'll have lower pool size. So far, this fits what we're looking at. Now, life gets more complicated when you're looking at glutaminase as the rate-limiting enzyme. Because when we inhibit glutaminase, what we expect to see in these very glutaminolytic tumors, we expect to see more glutamine in the cell because it's not being consumed, even though it's still being transported. And we also would expect to see less of the downstream product, glutamine. And this is an illustration from a cancer research paper that Rangju and our group and I had a couple of years ago. And so we're actually looking for an increased glutamine signal when we look at fluoroglutamine. Now, does this work? So from the Calithera group that developed the CB839 model, they did this assay in cells and breast cancer and found that when they applied their enzyme, they blocked glutaminase activity. They saw an increase in tumor glutamine. They saw a decrease in tumor glutamate, and they saw a decrease in some of the downstream products of glutamate, such as aspartate. Rangju repeated this using an NMR assay. And you'll see the letters HCC1806, that's a very glutaminolytic triple negative cell line, and MCF7, ubiquitous ER positive cell line, which is known not to metabolize glutamine. So when we give HCC1806, a glutaminase inhibitor, we see an increase in the tumor glutamine content. But when we do that to the MCF7, which note to begin with has plenty of glutamine around for biosynthesis, but is not chewing it up, we don't see much change, and we actually see a higher level to begin with. So we now take that into the clinic in our mouse models, and what do we see? Well, if we use, and this is a slightly different inhibitor, BPTES peptides, we see an increase in this uptake of the tumor. We didn't actually see much of that tumor by fluoroglutamine to begin with. When we stopped having it metabolize glutamine, we actually saw an increase in uptake, which can be seen as the change in the pool size. So it's fitting our hypothesis about what these kinetics might look like. And as a matter of fact, if we look at tumors that have high glutamine metabolism, we see low uptake to begin with and an increase, in this case using CB839. And if they have low glutamine metabolism in the ER cell lines, they're high to begin with and they don't change much with treatment. So by looking at the baseline activity and the perturbation with the inhibition of glutaminase, we can use this imaging technique to try to identify those glutaminolytic tumors. All right. Find the gas guzzlers that maybe are using the high octane stuff and the ER positives, but that may not be using as much of it. Now, I can't resist trying to make this into a kinetic argument. So what we're expecting is that this should be a largely reversible process and should fit a tissue reversible glutamine pool. It should not have a trapping constant K3, unlike FDG, where that's fairly significant. But we know that there's a small amount of trapping of fluoroglutamine probably in peptides as an object for biosynthesis. But that said, this fits largely reversible compartments. We fit this with a Logan plot, which is a graphical approach designed for reversible tracers, usually used in normal brain, but can be used in tumors as well. And in fact, what we see is an increase in the slope with glutaminase inhibition, showing that we're getting an increased pool size as we would expect. Now, if you've been watching the numbers, this pool size goes up by only 1.5, whereas our actual glutamine concentration went up by more like 2.2 to 2.5. However, there's just enough blockade of liver glutaminase that we actually get slightly higher circulating glutamine levels after CB839. And this actually matched exactly what we would expect based upon our biochemistry study. So interestingly, that the model fits the data, at least thus far. We're just starting to get into humans. I show you work from my colleague, Austin Pantel. Again, we see good uptake in this actual ER positive tumor. We have a triple negative, I think, that is being imaged this week. And you can see it doesn't quite fit the kinetics quite as well. There's probably a little bit of trapping due to biosynthesis, but not a bad job with a 1 or 2 compartment model. And thus far, what we see in the mouse seems to be holding in our early human studies. Now, here's something we haven't published yet, and we hope to publish soon. We've done this in abstract form. Many of you may recognize that this tracer called oxamin or flucycline that you may use in prostate cancer and brain tumors is actually primarily transported by transporters in common with glutamine. And in fact, we see this exact same effect in our mouse models with flucycline versus fluoroglutamine. And if anything, flucycline, which is a truly artificial amino acid, does not really have any peptide trapping. And so we're actually continuing to work on and validate this, but we may think that we may eventually do a lot of our glutamine imaging that we've been doing with fluoroglutamine with flucycline. That would be quite advantageous to us because we have an already approved tracer that's out there in the clinic and available that may help us through this. So just something to keep an eye on and something to think about when you're interpreting these flucycline scans. All right. So now the important question, does blocking glutaminase kill tumors? If we can now find these glutamine addicted tumors doing PET, does that kill the tumors? Can we knock out those gas guzzlers by getting rid of their high octane fuel? And the answer is, life's not that simple. That's the theme of my talk today. And as a matter of fact, this is hot off a press release that the Cantata clinical trial of CB839, the telanglistat, in patients with renal cell and other very glutaminolytic tumor, failed to hit its primary endpoint. And so the company remains working to try to identify the right setting for this. And in fact, we've been continuing to collaborate with them. They've been very good about supplying us compound with the idea of trying to find that same biology ourselves. So if this doesn't work, why doesn't it? Well, remember when we're looking at FDG PET, we're looking simply at the glucose metabolism and through glucose metabolism. And we would expect that a really hot tumor is using glycolysis and probably depending upon glutamine as its alternative fuel. But it turns out again that life is not that simple. And there is another fate for glucose that also is related to FDG uptake. And it's in fact in elegant work by Ralph DeBardenis's lab at UT Southwestern using isotope labels has shown that in fact, in lung cancer, when you get these really hot lung cancers that I had always assumed were purely glycolytic, they're not. They're actually aerobically using the majority of the glucose that we see making its way through FDG's 6P. So all these tumors, which we thought, oh, they need this high octane fuel to survive, they got plenty of high octane fuel if they use their glucose the right way. And I think we're now beginning to understand that there's more flexibility for those tumors that have at least a modicum of oxygen supply to be able to handle this. So this is part of the reason we're not killing these tumors with glutaminase as much as we would expect. So we now come back to carbon 11 glutamine to try to get some insights into what's happening. And for those that are familiar with carbon 11 chemistry, it is elegant in that you can look at the native substrate that's exactly the same as what your body's looking at. When you do carbon 11 metabolism, it's a huge mess because that down product goes everywhere, and it's hard for us to tell when we do a PET scan whether we're looking at carbon 11 glutamine or one of its downstream products. So let me show you a little bit about that mess that we've gotten ourselves into, which I think is beginning to bear some fruit. So one of the first things we look at is we see metabolites. I seem to get myself in this in just about everything that I do. And even after as early as 5 minutes, only about 50% of what's circulating in the blood is glutamine, and almost as much glutamate is there. So there's a lot of systemic glutaminase that is making glutamate in the blood. And by the end of the study, we're seeing more glutamate than we are seeing glutamine. Now, interestingly enough, when we look in the tumor, and the blue is the control and the red is the CB839 in the glutaminolytic tumor, to begin with, when we look late in the tumor, as you would expect, we see mostly glutamate. We've metabolized a lot of the glutamine that's been delivered. And when we inhibit this, we see less glutamate and more glutamine. Yay, that's just exactly what we were expecting because that's how we were interpreting our fluoroglutamine signals. And in fact, the kinetics don't look that different as we actually see a higher uptake in our early experience, and it's just our early experience, a little bit like what I showed you before, in the red line when we inhibit glutamine metabolism compared to baseline. However, there are some surprising results. One of the things that we're finding is that we can explain this largely by this rather complicated model. And we thought that a lot of what's going through the TCA cycle should be used for biosynthesis. And so we should be seeing a lot of trapping of this compound kinetically, but we don't. We don't see much K3 either coming from the glutamine a little bit or from the glutamate as we sort this out by looking at the metabolites. And we see a lot of glutamate coming in, and we see a lot of glutamate efflux. So what's going on? One of the early hints that we had that this was a little bit strange was that K1, that transport of blood glutamate into what we thought was a tissue glutamate compartment, is actually quite small. And that was a big surprise. And the bottom line is we're now hypothesizing that that tissue glutamate pool that arises from glutamine is not in the cytosol, but is probably in the mitochondria. And that's sort of the focus of some of our next series of studies. Now, what we were able to do is do a little bit more work and look at acid extraction and CO2 analysis. And basically, there's relatively small amounts of macromolecular, small amounts of biosynthesis happening from this. And there is a modest, but still relatively small radioactivity presence at CO2. The reason that's important is CO2 is the downstream byproduct of full metabolism in the TCA cycle. So it suggests that we are not really using this for biosynthesis. We're not really using this glutamine for energy. What's going on? And as a matter of fact, here Rangju made friends with our friend in mass spectroscopy, was able to find a C13 labeled glutamine in the same position that we had, and found a very surprising results. Of all the places that glutamine could go, it was stuck in the glutamate pool. So that these cells were burning glutamine like crazy to make glutamate. Remember, that's the next product after glutaminase. And in the MCF7 cells, they were not doing that. They were just having the glutamine sit there and presumably using a little bit more of it for biosynthesis. So the implications are, when we see in these kinetics glutamate in the cell that's glutaminolytic, a lot of it will come from glutamine and just sit there. And as a matter of fact, we have other studies that have shown this. I'll quickly show you some work that Rangju did with Ravi Reddy in our MR group, where he used CEST, chemical exchange saturation transfer, for glutamate, which he calls GluCEST, to look at this. And in fact, we know that glutamate goes down after the inhibition of glutaminase. And in fact, that's exactly what was seen in this early study of our same mouse model by GluCEST in a publication that came out by Rang about a year ago. And again, quantitatively see a good match between the GluCEST measurements and the measurements that we make biochemically. So we were kind of at a loss, and at about the same time, this study from Tim Whitney, who was at Stanford for a while, and then moved to Oxford in England, came out with a study using FSPG. Now, FSPG is a glutamate analog, and this is a glutamate analog that's not delivered the way we deliver glutamine, but it's delivered through the blood. And when you go through the blood, you go through this exchange transporter called the XCT, and this trades cysteine for glutamate. All right, a lot of the amino acid transporters that sit in the membranes are designed to be efficient and do exchanges. And as a matter of fact, although glutamine is a very abundant amino acid, these other things like cysteine and glutamate are a little less abundant. And so there's always a lot of trading going on between the cells and the plasma. Now, what he found in his ovarian cancer model is very interesting. When he gives sensitive ovarian cancer doxil treatment, and doxil is a rather oxidative derivative of doxorubicin, he sees a decline in FSPG uptake, which implies a decline in the glutamate pool, which is exactly what he found. When he takes a resistant model that is resistant to doxorubicin, he finds that at baseline, there's a smaller glutamate pool, and that smaller glutamate pool does not change with uptake. And it's a really interesting finding, but we were scratching our heads about trying to figure out what this was just about the time that the request to write an editorial on this paper came. And when we got this editorial request, and this was written by Sharon Lee, who runs our cyclotron, has been very active in the program, there was something buried in the online publication. That's why you should always read the online supplements, that showed when you deprived the cells of glutamine, you saw the same thing. So that at some level, that glutamate was coming from glutamine and was probably mediating this effect that we saw in these resistant cells. And so we put together this paradox, which we like to call the Lee paradox, but I don't know if Sharon's going to get that named after her, is that if we hypothesize that there's a mitochondrial compartment of glutamate coming through GLS from mitochondrial metabolism, and that pool is finding its way in and out of the XC transporter, this might explain some of the findings we have. And as a matter of fact, we know that glutamate has a number of antioxidative actions. And so it may very well be that the mitochondria's job, and what we're doing with all of this glutamine through glutaminase, is to actually make antioxidants for the cells. So let's go back to our gas guzzler that's using all of that glutamine. It needs fuel and it runs hot, and it releases all these oxidation byproducts. How does that relate to what we're seeing in our cells? Well, now we've redrawn this pathway to show that when you get through glutaminase, not only do you follow the purple line that we always thought was important to get into the TCA cycle and make energy and substrates, maybe what you need to do is send your pool of glutamate out into the cytosol so that it can refresh glutathione, very important in mediating oxidative stress, and also exchange with cysteine to be able to get the other ingredient you need to make glutathione. So if you were designing a system that said there's not much glutamate and cysteine around, but I still got to prevent the cell from oxidizing itself, this might be exactly what you do is, I'll burn that very plentiful glutamine, make glutamate on the bottom, and have it go out. So it's an interesting hypothesis that remains to be tested, but where we're going thus far. I will tell you, and we're not quite ready to show those data, that we are employing the strategy by blocking both glutamine and glutamate. And as a matter of fact, we're starting to kill highly resistant breast cancer cells in that fashion. More to come. This is the model that we're using to guide the studies, where we're now going to use a combination of flucycline and FSPG. We're starting to do that in our lab under the hypothesis that this is all about, or at least a lot of this is about, mediating oxidative stress. Okay, I have taken you through about 40 minutes now, and I probably made your head spin a little bit. If you're not a biochemist, you might have gone, what was he talking about? And if you are a biochemist, you might have gone, he doesn't know what he's talking about. So let me move on to something I know a little bit more about more solidly, and some of the new tools for the test. We've got these very complicated things going on. How can we help ourselves? So one of the things we can do is get better pictures that better reflect the physiology that we're looking at. So right now we take a picture, we draw a region of interest, we get curves, we do these kinetic models, but we've known for a long time that if we could actually make a kinetic picture, a picture not of simple FDG uptake, but of its delivery and flux through the system, this would lead us to advantageous work. A lot of people have been doing this work. Rich Carson has been, I think, preaching this for many years appropriately. And we've been doing some work in various reconstruction methods. Samuel Matej, who's part of our physics group, has the direct algorithm which can actually embed these equations into the reconstruction process. It's not a new idea, but it's actually a very efficient approach to Samuel. And you can see that not only can you calculate these constants by doing this, you actually can improve the quality of your imaging using this 4D direct iterative approach that constrains the biology. I've also been working for a long time with Finbar O'Sullivan when we were both at the University of Washington. Finbar's now in University College Cork in Ireland. He has taken a slightly different approach where he decomposes these time activity curves mathematically into a bunch of sum time activity curves using a mixture model, goes ahead and fits all those mixture models using a residual analysis, and then can make elegant pictures of otherwise very difficult to do studies. This is just an example of that flow, blood flow, glucose flux metabolism that I showed you before. And again, more to come. A couple of these are part of a moonshot grant that I'll tell you about in a second that we got for trying to develop these techniques. Now, I think one of the most interesting things we've done, and hot off the presses from the EJNM MI, is the idea of letting our computer scientists and Despina Contos and a very talented graduate student, Maria Chitalia, go at this. They went through 4D radiomics methods, and I'm not sure I completely understand this, but through a variety of techniques of segmentation, came up with an algorithm that efficiently segmented a phantom and actually started to produce results that we hadn't shown before. And she went back and looked at our historical data set from the University of Washington, which was about 50 dynamic patients, and pulled predictive value out of a baseline model that we were never able to do with standard imaging. Now that doesn't prove anything because it's just an exploratory process, but it suggests that when we can combine some knowledge of kinetics, maybe with some more modern computer segmentation and radiomics techniques in a very different fashion than you're used to hearing about AI and radiomics, we may generate some very interesting things. So this is an early iteration and more to come. Now, one of the things we recognize, and I showed you before, is how do we know when we have a glutaminolytic tumor? Well, it's low on fluoroglutamine, and it's high on FDG. And so we kind of need to do those scans at the same time. We actually need to do more than one PET scan to be able to recognize, starting with the glutaminolytic tumor and bringing it to a less glutaminolytic tumor after inhibition. And fortunately, we're in the position to be able to leverage our whole body PET scanner. We are part of the Explorer project. You all are also part of that as well. And we have the PenPet Explorer being designed complementary to the Explorer that has been developed in conjunction with a Chinese commercial company in Davis, and you'll soon be getting something, I believe, similar to that. Yale may already be there. Why whole body PET? Very high quality whole body images, captures whole body 4D kinetics, so you can understand this very complicated system biology, and it allows, we think, multi-probe capability because of its unique capabilities. This is just what our device looks like so far. It's actually just a couple floors away from me. It's up to 5 rings. It'll soon be up to 6 rings. And we're actually doing quite a few studies on there. Some of these in conjunction with the work that Bob Mack is doing with Rich Carson on a couple of joint projects. So what does this show you? Our standard Ingenuity takes a pretty decent scan in 20 minutes, still a little noisy. It's a lot noisy at 2.5 minutes. When we take that same patient and run him or her up to the scanner, we can see in a 16-minute scan, we get a beautiful scan. That's just over the slightly shorter field of view version of it. And in 2-minute scan, we get a better scan than we saw with our 20-minute narrow field of view scanner. So this makes a very pretty FDG PET picture. But there's more to it that you can do from that. Here's a dynamic scan of a patient imaged at 10 minutes and 1 hour 75, 4.3 hours, and 18.6 hours. Now, what does this mean? We see not much FDG uptake. Well, still getting to the brain, lots of FDG uptake in the brain, a little bit less, and nothing at 18.6 hours. So what's going on? Is that poor person had to listen to my lectures and just lost all of his brain power at the end of the process? Well, fortunately for him, no. But in fact, what we saw is that, as we would expect, the dephosphorylation of FDG, if we wait long enough and can measure it, because the brain uses glycogen, and using glycogen means you have to dephosphorylate sugar, and can also dephosphorylate FDG. We notice that the heart does not do that, and you see this heart at the end. We had the people cheat a little bit and go off diet and have cardiac uptake. And as a matter of fact, in the first study we did, we've already uncoupled enzymatic differences between the heart and the brain that we kind of knew about, but we never saw quite this dramatically. We can also get exquisite time activity curves. This is not an angiogram. This is a 3-dimensional PET study with pictures and the time activity curves to manage it. And again, this comes from Joel Karp's group, Varsha Visnowath, and Austin Pantel, and a few others working on this area. So this is where we are now with our Cancer Moonshot grant. We're proposing to do a fluoroglutamine study first, in part because it's not trapped. Then we go to a low, high dose FDG. We'd like to actually analyze both of those curves together using some techniques that Finbar Sullivan has developed, and generate a parametric approach that shows us exactly what we've been looking for, combinations to the glutamine pool size and delivery, and the FDG delivery and metabolism. So I'll stop here by telling you that glycolytic or glucose metabolism with FDG has had a profound impact on our cancer patients in our practice of nuclear medicine and radiology. But as complicated as it is to sit and interpret those PET scans on a daily basis, this biochemical interpretation may be even more complex than we think. So careful not to get fooled when you're looking at this. And I think more and more we're going to be able to look at other metabolic substrates, look at other metabolic substrates at the same time as glucose metabolism. And I think that's going to really have an impact on our understanding of cancer and our ability to direct cancer treatment, especially metabolic therapy. And so I close by showing you that not only can you measure what I've shown you from before by PET and a couple of tracers, but there are other important tracers. Carbon 11 acetate, I think, will play a key role in this at a branch point in the TCA cycle. We can use various MR-based techniques. Hyperpolarized pyruvate will delineate that branch point. We're already starting to look at that as well. And so I think this will be a multimodality effort with a very complex process, which I've literally been chasing for 20 to 30 years. So with that, I will acknowledge my partners and the grants that have partnered this, and some of the industry partnerships, Philips for the scanner, Calithera, and Blue Earth Diagnostics. And I will stop and actually leave perhaps a few minutes for questions. So I'll stop sharing, Miriam, and happy to take questions.
Thank you so much for an outstanding talk. We have quite a few questions already.
Okay.
So Dr. Darko Pukar from Nuclear Medicine says, great talk. Please summarize potentially clinically applicable agents targeting tumor metabolism. When Craig Thompson became Memorial Sloan Kettering president, I expected much faster clinical oncology translation.
Yeah, that's a good good question. I expected to be doing what I just showed you many years before I got to. So one thing I'll say is that metabolism is incredibly complex. And if we could find another FDG, it would get a little bit easier. And that really hasn't happened yet. And as a matter of fact, we differ a little bit in Sloan Ketter's interpretation of fluoroglutamine. Mark Dunphy, who's done some outstanding work there, thinks that some of what he's seeing in patients is fluoroglutamine metabolism. We actually don't think so. And we're starting to have a hypothesis that shows this true. The other thing is, I'm going on a little bit on a limb here based on our preliminary data, but the interpretation of what's happening and what we did with tracers, I think might change the interpretation of what's happening with glutamine. Because the classic line is that glutamine is used for energy and biosynthesis, and there is definitely a recognition that it's used as an antioxidant, but at least in our triple negative xenograft models, that looks pretty important, and it looks pretty important to overcoming resistance. So it's a very long-winded way of saying metabolism is very complicated. And I expect that we're going to see more metabolic substrates. I think better metabolic agents will drive that, and those two are interdependent. So I think these metabolism tracers are difficult. Will take some work, and I think we got pretty lucky with FDG.
Thank you. We have another question from Dr. Rob Goodman, who's our department chair. When you design your studies on novel PET tracers, are you required to generate, fund, or use a minimum number of doses in order to make the novel tracer generation process cost effective?
Yeah, no, my day job as vice chair, my former job, now taken over by Dan Prima as pet center director, we always had to worry about the costs. And it's a tricky process. Looking around the country, number one is this is expensive and it needs grant funding. And so we have a pilot program that will generate just enough data to get grants. And that's a part of what we do. We also, and through the great contribution of our department and our chair, Mitch Snall, long before I ever got here, have a joint venture going on with a hospital who realizes that for us to stay at the cutting edge of this very competitive field like PET, it helps us to do research. And that in fact has borne fruit both from a research and a clinical revenue standpoint. We actually make our own FDG, not because we can't buy it somewhere else, but there we can actually charge for our FDG and use all the personnel that we need to make FDG by FDA compliance, who are also needed to make these investigational tracers using FDA IND compliance. So we leverage our costs to whatever extent is possible, but these programs need center grant funding, they need clinical revenue, and they often need some support beyond that to make them profitable. I know there, Rich has done a fabulous job of engaging commercial support that utilize the scanner and collaborate with him, and I think that's been an outstanding model. But everybody's got to figure out how to pay for these things, and we're all still working on it.
I did want to ask you, in terms of the tracers, amino acid tracers that are available to us, not FDA approved yet, besides Oxymin, which I guess, which one, there's been a lot of talk about FET and FDOPA, and
Yeah.
which ones should we be focusing on, or just focusing on the one that's available?
Yeah, here's where I want to separate the brain tumors and everything else. And the reason for that, and I actually think that's why we got a little bit different results from Sloan Kettering, because we started right with somatic tumors, and they've been looking at they looked at brain tumors at least early on. In the brain, these amino acids that you just mentioned have a really nice property, is they will get across the blood-brain barrier because the tumor has in association with it an upregulation of amino acid transporters, and so you can see uptake without blood-brain barrier breakdown. And as a matter of fact, flucycline, which I think is probably going to be taken over by PSMA agents for the prostate, I think is going to find a home in brain and other low-grade tumors. So all of those things that you just mentioned have the commonality of high-grade uptake, including in low-grade tumors. And so they're really good at delineating tumor margin, and it's different than contrast enhanced MR. And I think in particular, one of the reasons these glutamine analogs work so well is it's kind of the inverse. You may actually get more uptake when you have a lower grade tumor, so it may actually be quite good at looking at the low grade tumors. In the rest of the body, it's tricky because you're looking at a combination of amino acid transport and washout, and anybody who's done a flucycline in the prostate knows you can't wait too long until you image that. And I think as we understand that and perhaps adapt ourselves to trying to measure these distribution pools, we'll be able to use it. So I think, and again, I'm not an amino acid expert other than having to delve into the glutamine piece of it. I do think flucycline is going to have some utility beyond prostate cancer, and a lot of it will be for these other tumors that are not FDG avid. And we think the reason they're not FDG avid is because they're using amino acids, but they're not burning them, like the high grade, highly FDG avid tumors. How's that for a very long answer to a short
I love that answer. I love that answer. Do you think flucycline will have a role in breast cancer imaging?
Yes. And I sure hope our grant reviewers do too.
Okay.
Yeah, there's been some good work in it. Gary Muehllehner, who was at Memorial Sloan Kettering, who's now out at the Hoag Center, published a study and showed actually you see surprisingly high uptake in lobular breast cancer, which does not surprise us now, based upon what we've seen, because we know they're not particularly glutamine metabolism. So I think it's beginning to think that at least in the body, those places you see flucycline working well, low-grade prostate cancer, low-grade breast cancer. It's been shown in some of the other low-grade tumor types more incidentally. It's beginning to make sense. These retain their amino acid transport probably for biosynthesis, but are not chewing up those amino acids as they come in. So yes, I think you will. I think Blue Earth is likely to have, as you know, probably as well as anybody else, an indication for brain tumors relatively soon. I'd like to see it tested for breast cancer. We're actually proposing to do that in our animal models. We've got to complete our fluoroglutamine study, but we may switch over to flucycline for the next run.
That's really exciting. Yeah, we're running the brain mets trials with flucycline and seeing really exciting results. You know,
and as you know, we are too. I've got to go convince Ali, who's running those here, that that he's looking at more than just uptake. But I think he's buying into it. So.
Yeah, well, Ali actually has started talking to us about collaborating with to extend this to pediatric brain tumors.
Yeah,
so
I think that's a great idea.
We're excited to join force with him. So.
Yeah, there's a huge interest to CHOP there, and so keep that going, please.
Yeah. Any other questions? Because I can talk about amino acid PET for a very long time.
As can I, without knowing much about it. Interestingly enough, I just came from a talk that John McConaughey was running for ECOG.
Oh, yes.
He's an expert in, so you'll have
course.
to invite him up for true amino acid experts, but I'll pretend I am.
We'll try to invite him. We did invite him to ASFNR, so we're excited for that. Great. So, well, thank you so much. This was a fantastic talk. I learned a ton. I finally understand why oligodendrogliomas take up so much.
At least as far as I can tell from breast xenografts, right?
Exactly. But I'm so grateful for your time, and I know we all learned a lot.
Well, thank you. It's a pleasure to come back to Yale virtually and in person, and I look forward to the time when we can do this all together, and I can actually see those libraries that the folks are sitting behind you on the screen. And it's a real pleasure. I have to say that, as you know, it's really been a delight to have the Yale PET Center is probably our most important partner right now. I think Rich may spend as much time Zooming with Bob Mack as he does anywhere else. And I'm really delighted to do that. I'm really delighted to be invited back. And I hope the next time you come back, I finish the rest of the story and that I didn't have to reverse what I just said, which I don't think so. I did leave a copy of my slides. I took out some of the preliminary data we haven't published.
Yes.
Some of the stuff that's been published, I'm happy for you to share that with everybody.
Yes. Thank you so much. We're excited to continue collaborations and learning as much as possible.
Thank you, and I appreciate the invitation.
Thank you.